Good afternoon, everybody. Welcome back. Today we're going to cover a topic called transformation and oncogenesis. We're going to start with an experiment where we take a hamster embryo. We're going to chop it up and treat it with trypsin to make single cells, and we're going to plate them out in 2 cell culture dishes, cover them with medium. And one of them we're not going to do anything to. We're going to let it grow out, and you'll see that in these cell cultures, if you look at them under the microscope, eventually you'll get a nice monolayer of cells, or a cell sheet as we call it, where the cells grow and divide until they touch each other and then they stop. And the other one, we're going to treat the monolayer of cells with a chemical that causes mutations. And in those monolayers, in that monolayer, we're going to see foci of what we call transformed cells. We can see that they are very different morphologically in the microscope. When we look at them close up down here on the lower right, you can see they're morphologically altered, and they're also piling up on one another. They don't show what we call contact inhibition. They don't stop growing when they touch each other. So that's a transformed cell. And that process by which these cells become transformed is the topic of much of today's lecture. And on the right are 2 graphs illustrating this in a quantitative way, where we are looking at, on the top anyway, mouse cells. We're looking at the cell growth rate with days after initiation of the culture. So if you did this experiment with a mouse embryo, you plated out cells and simply didn't treat them but let them grow and split them every few days, you would see that within 20 days or so, well, first of all, the number of cells go down, and within 20 or 30 days, they undergo a crisis where most of them die. So on this decreasing curve, the cells are dying. And then a few cells remaining will grow, populate the culture, and those cells are immortal. And they're transformed just like the cells, the hamster cells were that we treated with a mutagen. Now, on the bottom is what happens with human cells. You can plate out human cells, and you can split them for a number of generations. That's on the x-axis here, so slightly different from the above graph. You can see they'll grow for a while, about 50 doublings, and then they die and they crash. And so it's very hard to make immortal or transformed human cells. It can be done. But I want to explain the difference between these 2 cultures, and in so doing, we'll explore a little bit what's going on in transformation. And this all has to do with telomeres, telomerase, and cell immortality. Telomerase is an enzyme at the ends of chromosomes. It's active in all cells during early embryonic development, but most somatic cells, most mammalian somatic cells, do not produce an important enzyme in telomerase called TERT, which is a reverse transcriptase. Well, this all has to do with the ends of chromosomes. Each chromosome has an end that has an end problem. Every time the DNA is duplicated, there's a gap and you can't fill it in. Not clever like viral genomes and get around the end problem, but there's a gap. And so this has to be protected. Otherwise, the ends get shorter and shorter and they get degraded. In particular, the DNA damage response of the cell would see it and chew it up. So there's a whole bunch of proteins, telomeric proteins, that bind to telomere ends to protect the ends. But then there's also a telomerase, a reverse transcriptase that copies an RNA, a short RNA, telomeric RNA, to maintain the ends during early embryonic development. But once you're born, your somatic cells no longer produce TERT, so the ends get shorter and shorter. Now, in an organism, it doesn't matter because cells turn over. Most of our cells turn over and are replaced. But in culture, every time you split the cells, or every time the cells divide, actually, the telomeres keep getting shorter and shorter until about 4,000 bases, the cells die. They induce apoptosis, and that's why the human cells only survive about 50 divisions, as we showed in the previous slide. It turns out that mouse telomeres are longer. Their fibroblasts in culture can divide for more generations, and that increases the likelihood that spontaneous mutations are going to make them immortal. And that's why we can derive immortal mouse cells in culture in the previous experiment, because their ends are longer. So these kinds of mutations that lead to immortality, they can be in a variety of genes, as you'll see in a moment, but some of them could increase telomerase production, turn it back up, and those cells would be immortal. So that's what's going on there in cell culture. A summary of what a transformed cell is. And when people first started observing these years ago, when it became possible to make cell cultures in the first place, they were puzzling. People didn't really understand why there were normal cells and why there were transformed cells. But a few of the properties that are important for our discussion today, these cells are immortal. They will grow indefinitely. As long as you keep feeding them and splitting them in the laboratory, they will keep growing and growing and growing. And a HeLa cell line is an example of that. And we'll get back to that at the end here. These cells have lost anchorage dependence. They don't have to be attached to the plastic any longer. They can pile up on top of each other, which is also contact inhibition. Normal monolayers in panel A on the right, they touch each other. The cells touch each other. They stop growing. They don't pile up, but transformed cells pile up. These transformed cells will also make colonies. If you suspend them in agar and plate them out, they will make colonies, whereas normal cells can't handle that. And they have decreased requirements for growth factors, including serum. They need less serum. A transformed cell line needs less serum than a primary cell line, which is made from a tissue. Now, this property, transformation, is different from oncogenesis. That's why this lecture is called Transformation and Oncogenesis. They're two distinct properties. Oncogenesis, of course, is the development of cancer, and a tumor is a swelling caused by abnormal growth of tissues. It can be benign, stays restricted, or it can be malignant and metastatic and spreads elsewhere. And cancer is a genetic disease. About 8 million deaths per year in developed countries. And we have learned that cancer is caused by about a dozen mutations in 12 different genes, affecting signaling pathways that govern cell proliferation, survival, cell fate, and maintenance of genome integrity. And so you could inherit some of these mutations from your parents. And then when you get up to 12, you develop a cancer. You may not do that in your lifetime, but many people develop cancers. And in your lifetime, you may acquire additional mutations, DNA damage caused by environmental carcinogens, infectious agents, ultraviolet light, random mutations that don't get corrected. And so we think about 12 are needed to make a cell have all the properties, not just to be transformed, but to be a cancer cell. And what we're going to see today, just a preview, is that viruses make cells transform. They make them divide forever. Dividing forever is the problem, because with each cell division, mutations are sustained, and eventually you get those 12, and the cell can become oncogenic. All right, so that's the key to this. A cell normally doesn't last very long because of this problem, because if it keeps dividing forever, it just sustains mutations. So transformation and oncogenesis are distinct properties. Transformation is this property defined in cells and culture. Normal cells, transformed cells, we talked about the different properties. The transformed cell may or may not be oncogenic. You could take these transformed cells and inject them into a mouse without an immune system, a nude mouse, for example. It may or may not form a tumor. This tumor formation requires additional genetic changes from the ones that are in the transformed cells. All right, transformation, oncogenesis are distinct. And the reason we understand all of this today is because the study of virus-transformed cells gave us insight onto what happens in cells to establish oncogenic potential. What is it that makes cells divide forever? And now that we're studying cancer cells, what mutations lead to cancer? The key here is that no virus can do it all. A virus can transform cells. A virus doesn't cause cancer. A virus causes a cell to become immortal, and the making of the cancer is up to the cell itself. It's up to whether those 12 mutations are sustained during that uncontrolled replication. So viruses start the process, and then the cells take over. We know of these human cancer viruses. There are others that cause cancers in animals, but this table is just for human cancer viruses. There are RNA viruses like human T lymphotropic virus 1 that causes leukemia. HIV causes a variety of cancers in different tissues and organs. We'll talk about that later. Hepatitis C virus, of course, liver cancer. And then there are DNA viruses, Epstein-Barr, Kaposi sarcoma herpes virus. These two herpes viruses causing a variety of human cancers. Hepatitis B virus, of course, liver cancer. Human papillomaviruses, cervical, penile, anogenital, head and neck tumors, and Merkel cell polyomavirus, a skin cancer. These viruses are a contributing factor in about 20% of human cancers. And the ways that they cause cancers are different. And in fact, we're not going to consider them today. The only relationship would be to HTLV1 causing cancer, as you'll see in a moment. What I'm going to do is tell you a story about how we learned how virus infections led to cancers and how that elucidated the mechanism of cell growth control, so that now we understand how these viruses cause cancers, how cancers are caused by other effects, non-viral effects as well. What I do want you to remember, especially, is that transformation in oncogenesis, virus-induced cancer, is not required for the replication of any asterisk virus. There's one exception as far as we know, and that is a virus called wall-eye dermal sarcoma virus that causes tumors in fish, in walleyes. And there it is right there, the tumor on this fish. This is the only example where the tumor is actually needed for virus transmission to a new fish. These fish develop these tumors in the water, of course. They fall off. The fish is fine. It lives on and never gets another tumor, but the tumor is full of virus, and when it falls into the water, it breaks up, and the virus spreads to other fish. For all other examples that we're going to talk about today, transformation and oncogenesis are not needed for their replication. They are accidents. Yet for us, studying them, great accidents, because it taught us a lot about cell growth control. This begins in 1909. Peyton Rouse, a scientist at the Rockefeller Medical Institute, that's what it was called back then. Now it's, of course, Rockefeller University. A farmer brought him a chicken with a tumor. It's right here. See that big mass on the outside? That's a tumor. It's an English hen. Farmer brought it to him because Rouse was interested in understanding what was causing this tumor. So he took off the tumor, he ground it up, he filtered it. So by now, 1909, we know about filterable viruses. And he injected the filtrate into another chicken, and that chicken developed the same kind of tumor. This was a sarcoma, a solid tumor. First time showing that a solid tumor could be caused by a viral infection. Actually, leukemia had the year before been shown to be caused by viruses, but no one thought that was an actual cancer. So Rouse got the credit for that initially. Took 50 years for the world to believe him. In 1966, he got the Nobel Prize for this work, the longest incubation period of any Nobel Prize. Why? People didn't believe him at first. Other people worked in the field. They messed it up, all kinds of reasons. But eventually, as you will see today in my story, the world came around to understanding that, yes, he had found the first virus to cause a solid tumor. And his virus was called Rouse sarcoma virus. Rouse, his name, sarcoma, that's what it caused in the chicken. And you will see, in addition to his Nobel Prize, 2 other Nobel Prizes were awarded for working on his virus. And I want to tell you the story of that in the beginning of today. And I'm going to tell it to you by reading a few passages from this wonderful book, The Emperor of All Maladies by Siddhartha Mukherjee. He's won a Pulitzer Prize. He is an oncologist here at Columbia, actually. It is awesome. I'm sure some of you have read this. If I were in front of you, I would ask, and some of you would raise your hands. But anyway, tells a great history here. By 1950s, cancer researchers had split into 3 feuding camps. Remember, 1909 was Rouse. The first, led by the virologists led by Rouse, claimed that viruses cause cancer, but nobody could find a human cancer virus. Epidemiologists thought that chemicals caused cancer. They couldn't find a mechanistic explanation. And so these chimney sweeps in England, they would wear shorts. They were little kids, and they would wear shorts to get down the chimneys, and they got testicular cancer by exposure to the tar and other chemicals in the soot. Great epidemiology to point it out, but they didn't know how it would work. And the third camp possessed weak circumstantial evidence that genes in the cell might cause cancer. In the end, these were all right, and it all comes together, as you'll see. In comes Howard Temin, 1951, young virologist. He goes to Caltech to study fruit flies, but he got bored with fruit flies, and he decided to go to Renato Dulbecco's lab and study Rous sarcoma virus. So here's Renato Dulbecco. You may remember he developed a plaque assay for animal viruses. He recognized as soon as cell cultures were developed, this would be good to study viruses, and he thought he could study Rous sarcoma virus in chicken cells. Up until this time, the virus was only studied in chickens. People would inject the virus into chickens, they would get tumors, and that's it. They never could figure out any mechanism. You need cell culture to do that. But Temin wanted to do it in cells. He wanted to create cancer in a dish. These are the words of Mukherjee. And he did it after 7 years in Dulbecco's lab. He added Rous sarcoma virus to a layer of normal cells in a petri dish. The infection incited them to grow uncontrollably, forcing them to form tiny distorted heaps containing hundreds of cells that Temin called foci. These, he said, represented cancer distilled into its essential elemental form, cells growing uncontrollably, unstoppably. Now, this is not cancer. This is transformation. Temin found that he could transform cells in culture with this virus, with Rous sarcoma virus. And here are two examples of transformed cells. And they can have different morphologies. On the left, fusiform. There's a focus in the middle of transformed cells. The normal cells around it, and these are round and refractile transformed cells. But in both cases, what's happening is you have mostly normal cells, and then some of them infected by the virus, and they're rare. Not every cell happens. Even though maybe all these cells were infected, the cells are transformed and they piled up. So from that discovery, Temin said, this is an RNA virus permanently changing the cells. It's transforming them. He said, this can't happen with an RNA virus. It must be that this virus changes its RNA to DNA. And that led him to discover reverse transcriptase and Rous sarcoma virus. And he in Baltimore got Nobel Prizes for that. So Nobel Prize #2 for Rous sarcoma virus. And then we have some DNA viruses that are shown to transform cells. In 1962, Polyomavirus found to transform hamster cells. As you'll see, this is a mouse virus transforming hamster cells. 1964, Simian virus 40, a primate virus transforming mouse cells, 3T3 cells, or mouse cells. And again, just like Rous, most of the infected cells died, but rare cells did not. They were transformed. They had the properties that we just described. So we have RNA and DNA viruses transforming cells. How can this happen? Well, you have to reduce cytopathic effects, reduce or eliminate them. Otherwise, you would kill the cell. So transformation requires the infected cell not die. You have to reduce virus replication so that virus particles are not made. And the cell must continue to divide. It becomes immortal. Does this ring a bell? Do these properties ring a bell? This is a persistent infection. It's a very specific kind of persistent infection where the presence of the virus has immortalized the cell. But all these properties are very reminiscent of what we talked about last time, persistence. Which of the following is not a property of transformed cells? A, increased requirements for growth factors. B, immortality. C, loss of anchorage dependence. D, loss of contact inhibition. E, colony formation in semi-solid media. All right, let's see how we did. That's right. Most of you got A, increased requirements for growth factors. That's wrong. It's not a property. They have decreased requirements for growth factors. Immortality, loss of anchorage dependence, loss of contact inhibition, colony formation, and semi-solid media. Those are all correct properties of transformed cells. So now what I would like to do is tell you the story of how we used a study of viral transformation of cells in culture and how we related that to cancer. It was a convoluted pathway. It involves the study of retroviruses, it involves the study of DNA viruses, and some in vitro cancer biology. And these three fields converge in the 60s and 70s. And the end is that we have our current day a unified theory of cell growth control, mainly from the study of these viruses. It's really a remarkable story. So let's start with the retrovirus part. How does Rous sarcoma virus cause tumors in chickens and transform cells in vitro? And let's talk a little bit about Rous sarcoma virus. It turns out that its predecessor, Avian leucosis viruses, these are endemic in virtually all chicken flocks in the world. So here is a Rous sarcoma-like virus or an avian leucosis-like retrovirus. These are envelope viruses with envelope glycoproteins. They have a capsid which contains 2 copies of the RNA genome. And of course, the particle also contains reverse transcriptase and integrase and protease. These viruses, ALVs, were discovered by Ellerman and Bagg in 1908. They found that they caused leukemia in chickens, but remember, people didn't think that was a cancer. Most chickens are infected with ALV within a few months of hatching, and leucosis or leukemia, so leucosis is another word for leukemia, occurs sporadically in infected birds when they get older. About 3% of older birds develop leukemia. It's 14 weeks of age. Not many birds live that long. I guess the ones that lay eggs do. But a few percent develop leukemia sporadically. We'll actually explain later why they do that. The other 90% of the birds have a transient viremia. They get immune and they don't get leukemia. And that's the end of that. So every chicken you eat, every egg came from a chicken that at some point was infected with an ALV. I bet you didn't know that. Now, these infected birds, the 3%, as they get older, they develop solid tumors like the Rous sarcoma. So a sarcoma is a connective tissue tumor. And Rous got viruses from his tumor and showed that they rapidly cause sarcomas, not leukemias. So this bird was a birth infected with avian leucosis virus, and it may or may not have developed a leukemia, but it certainly developed a sarcoma. And that's what Rous isolated his virus from, and he called it Rous sarcoma virus, of course. And many other people did this subsequently, and they isolated their own sarcoma viruses of chickens, but they turned out to be defective. I will explain what that means in a minute. I just want to put up front here that Rous was very lucky. His virus was not defective. And if it were, it may have been a problem for him, but he was very lucky, and he had a non-defective isolate, whereas many other people had defective ones. So how does Rous, but not ALV, cause sarcomas? Avian leucosis virus causes leukemias. Rous causes sarcomas? Well, this was studied by a number of people, and a key finding was that the genomes that you could isolate of viruses from solid tumors like RSV were recombinants. A piece of the gene, so they're basically derived from ALV. Rous started off as ALV. A piece of the genome is replaced with a piece of host DNA that was called an oncogene. And these were studied enabled by molecular techniques in the 70s. Michael Bishop and Harold Varmus identified the oncogene present in Rous sarcoma virus in 1976. It was called VSARC. And they got the Nobel Prize for that in 1989. Third Nobel Prize on Rous sarcoma virus. And here's Mike Bishop and Harold Varmus. And you may know Varmus was head of NIH for a while. He's now at Sloan Kettering here in New York City. I interviewed both of them separately. So Harold Varmus came up here to Columbia for TWIV 400, and I went to San Francisco to interview Mike Bishop. He was out at UCSF. So they discovered that this virus, RSV, is recombinant. It's got a piece of host DNA, which they called an oncogene. So what's the major insight? So birds infected with ALVs, they get a variety of tumors. And if you take different birds, you can isolate different tumors from different birds. They all contain retroviruses derived from ALV, but most of them are defective. They're all different, and they've picked up different oncogenes. And as I said, Rous was lucky. His isolate was not defective. All of these isolates from these solid tumors of birds, they had different host DNA, not the Sark gene found in RSV. So you could go to Texas and do your own experiment with a Texas chicken and find a sarcoma with a different isolate that's picked up a different gene. Each DNA segment had a novel chicken oncogene. Amazing goldmine for molecular oncology. This really taught us how cell division is controlled. So here's a map of some of these, what we call transducing retroviruses, because they pick up pieces of host DNA. So here on the left, we have our avian transducing retroviruses. And here's avian leucosis virus, the virus that infects every chicken in the world and causes leukemia, gag, Pol envelope genes. And then below are all the viruses isolated from chicken tumors. Rouse sarcoma virus. It's got the Sark oncogene stuck in at the end of the genome, so it's not defective. It's got gag, Pol, and envelope. So this virus can replicate on its own. But look at this second one, avian myeloblastosis virus. It's got an oncogene called cMIB stuck in it. It has inserted right into the envelope gene. There's no more envelope here. This virus cannot make particles on its own. It needs a helper virus, so it's defective. And all these other viruses isolated and identified different oncogenes, ETS, MIC, MIL, YES, ERB, REL. They're all mostly defective because the oncogene is stuck somewhere in the retroviral genome. So that's what I mean when I say Rouse was lucky. There are also mammalian transducing retroviruses. People said, there are cancers of mice. Murine leukemia virus. It's a leukemia virus. Gag, Pol, envelope. They could then isolate different kinds of viruses from mice, other leukemia viruses, sarcoma viruses, and even cat cancer viruses and simian cancer viruses. They all have picked up an oncogene from host cells, and they're all mostly defective. And all these oncogenes with these names are cell genes that have been picked up and enable these viruses to transform cells, which then eventually become tumors. The virus just does the transformation part. Becoming a tumor is up to the cell to sustain the number of mutations needed to make a cancer. So let me just emphasize defective versus non-defective. A defective virus needs a helper. So if you have one of those transducing retroviruses that's defective, you can't grow it unless you mix it with a helper virus, which is basically the parent that has all the genes that it needs. And so the example here, avian leucosis virus, Rouse's virus, Rouse sarcoma virus, is non-defective because the oncogene went in addition to the genome. But avian myeloblastosis virus, BA1, is defective, and you cannot grow this without ALV as a helper virus. So that's what I mean by defective versus non-defective. How do these viruses pick up an oncogene? Here are two ideas for oncogene capture. These viruses, of course, make proviruses. They integrate into the cellular DNA in a relatively random fashion, and occasionally they integrate next to a cellular gene that controls growth. We call those oncogenes, although it's not a great name really, because in the cell they're not there to make cancers, which oncogene implies. They're there to regulate growth, but they were discovered because they're involved in cancers, and so the name has stuck. But this particular retrovirus is integrated next to an oncogene, and so that virus will pick it up in one of two ways that are shown here. First of all, the right-hand LTR could read through. So remember, the viral genome is made by a promoter in the left-hand LTR, and it usually terminates in the right-hand LTR. But sometimes the termination doesn't work. You get read-through, and now you make a transcript that includes the oncogene. That can get packaged into a virus particle, and then during reverse transcription, when you infect a new cell, copy choice will give you a new viral DNA with the oncogene in it, and this is now a transforming retrovirus. The other way that this can happen is sometimes the right-hand LTR is actually deleted. Recombination occurs in chromosomes, of course. The right-hand LTR is deleted, and you get a transcript from the first LTR read-through that contains the oncogene, which is in red, packaged in the particle, reverse transcribed, and then you deliver that oncogene to the cell. So the key here is now we have this oncogene as part of a retrovirus. It's now under the control of the LTR, which is really unregulated. LTR doesn't respond to anything. It's just pumping out mRNAs of the virus all the time, and so making these oncogenes in an unregulated fashion is not a good idea, and that's what transforms cells. So these oncogenes are all present in cells. The retroviruses pick them up. In the cell, we call them proto-oncogenes. There are over 60 that have been identified. They're in all cells. They control cell growth, and they are highly regulated because you don't want cells dividing all the time. You only want them dividing when you need them. If they divide all the time, they'll get mutations in their genomes, and that'll cause cancers. And the normal cellular versions of these genes are called C-oncs, like C-sarc, C-mix, C-mos, C-ras, etc. The retroviruses, when they pick up these genes, they're called V-oncs, like V-sarc, V-mic, etc. So sometimes the viruses pick up the normal cell gene and they just overproduce it, and that's why it transforms the cell. But sometimes in the process of being picked up by the virus, the oncogenes sustain additional mutations that make them even more potent in terms of activating. And the way these work, as you will see, is that they act at the G0 phase of the cell cycle, and they're like a green light for going through the cell cycle. These oncogenes tell cells to progress through the cell cycle, to divide, and they just do it continuously, and that's why they're transformed. So all of these oncogenes, 60 oncogenes or so, fall into this growth control pathway. And here on the left, there's some transducing retroviruses of different sorts, isolated from various species, as you can see here, birds, cats, mice, even simian ones. And then on the right is the encoded protein where it works in the cell growth control pathway. And some of these oncogenes are actually encoded ligands for growth factor receptors. These are growth factors that bind to receptors on the cell surface. So this growth factor would be something that you add to a cell, it causes the cell to divide. That's what a growth factor means. So some oncogenes are actually growth factors themselves. Some are the receptors, some are the parts of the signaling pathways that bring the signal from the surface into the cell, and some of them are even transcription factors that turn on genes that make cells divide. So remember, the original phenotype of these transforming retroviruses in cultured cells was transformation, uncontrolled cell growth. What they're doing, they're delivering these various proteins to cells in an uncontrolled fashion, and when you deliver any one of these to a cell, it makes the cell divide. And the virus is there permanently. Remember, it's a provirus. It's integrated. These proteins are made continuously. That's why the cells divide forever. And if this is in a person, a chicken, a mouse, it's going to cause a tumor eventually, as that cell sustains the other 12 mutations needed to cause a cancer. All right, so now we have to talk a little bit about the cell cycle. And we're going to get into this more when we talk about DNA tumor viruses. But again, the cell, about a 24-hour cycle. These proto-oncogenes, these cell genes identified in transforming retroviruses, we call them dominant oncogenes because you just need to put one into a cell to make it transform. This was all revealed by studying these transforming retroviruses. We had no idea how the cell cycle was controlled. Now we know that up here at G0, there are all these genes in a pathway that can turn on cell division, and we know that because we've isolated them in these transforming retroviruses. So retroviruses end up transforming in three ways. I've just told you about one, which is rapid tumor formation like Rous sarcoma virus, about 2 weeks. The virus has picked up a dominant oncogene, and that protein is made immediately when the virus infects the cell. It's permanently produced because the virus integrates as a provirus and the cells become transformed. There are retroviruses that transform with intermediate kinetics. It takes months, and this is avian leucosis virus, right? Remember, the chickens initially get infected, but it takes a while for them to develop leukemia. These viruses do not pick up a oncogene. All right, these are not transducing retroviruses. Rather, they integrate next to an oncogene and they turn on its expression. They can turn it on via the promoter, the right-hand LTR, or even by the presence of an enhancer sequence in those LTRs. So that's why ALV takes some time as to get into the genome, turn on the oncogene. It takes time for the protein level to build to the point where it will turn on cell division. And then we have viruses, retroviruses, that take years to cause tumors. Here's a human, HTLV1. There's no oncogene, doesn't cause cis activation, but rather a viral regulatory protein activates oncogenes by transactivation. So let me show you a picture of this to make it clear. So we have transducing, rapidly transforming retroviruses like Rous sarcoma. They've picked up an oncogene. We have the intermediate months like ALV, cis-acting, where the provirus integrates next to an oncogene and the right-hand LTR turns it on. That's one mechanism. And then we have the slow transforming, where there's no oncogene, there's no integration next to an oncogene, but rather the virus encodes a transcriptional activator. Here it's called X, and X protein is needed to activate the LTR. And that transcriptional activator can also activate a cellular gene, which, when activated, causes transformation. And one example would be IL-2. This can turn on IL-2, which will make T cells divide. This is a leukemia virus, and so that's explaining why those types of cells divide. These mammalian transforming retroviruses, they transform cells as a mistake. It's a byproduct. It's a byproduct of their having to integrate their DNA into the host chromosome. If you integrate into the wrong place, you're going to transform the cell or pick up an oncogene. It's a mistake. The virus absolutely does not require those oncogenes. In fact, in one of the first cool experiments done with Rous sarcoma virus, they showed that you could mutate that oncogene, the cell, and the virus still replicated quite well. You don't need the oncogene to replicate. It's a mistake. It's a good mistake for us because we learned about transformation in cancer, but the virus absolutely not needed. No requirement for transformation or for cancer causing in the reproduction cycles of these viruses, with, of course, the exception of walleye dermal sarcoma virus, which requires tumor formation because the tumor falls off the fish and it spreads the virus in the waters to other fish. Here's another question. Which of the following allows Rous sarcoma virus to transform cells? Presence of the envelope gene, presence of a Pol gene, presence of a Sarc gene, presence of LTRs, none of the above. Rous sarcoma virus. What allows Rous to transform cells is the presence of a Sarc gene. Picks up a Sarc gene. Now, a lot of you said LTRs because you're thinking it's integrating next to an oncogene, but that's not how it works. It actually picks it up, and that allows it to transform. Presence of LTRs allows the type 2 transforming viruses to transform cells by integrating next to an oncogene. All right, so that was our contribution of retroviruses. We'll come back to it, but now I want to tell you how the study of DNA viruses transforming cells revealed more about the regulation of the cell cycle. This starts in the 1920s with DNA viruses and converges with the retrovirus studies to give us a current unified theory of cell growth control. This is a different story. First oncogenic DNA virus was a papillomavirus that causes warts. A papilloma is a wart in cottontail rabbits. This was identified in 1933 by Richard Shope. And this is these are rabbits with papillomas, big warts. You can see growing out of their head and their lips. This one is particularly angry looking, I think, on the right. These are actually harmless. They fall off, but they're caused by infection with a rabbit papillomavirus that spread from rabbit to rabbit. If you've ever heard of a jackalope, this is like a mythical creature. You can look it up, Google it, you'll find it. It's supposed to be that people see this in the woods, a combination of a jackrabbit and an antelope. Well, there's no such thing. This is all it is. They see these rabbits with these big papillomas and they think they're horns, and that's where the jackalope came from. Anyway, this is the first DNA tumor virus discovered. Remember, papillomas are small DNA circular viruses, very much like polyomaviruses. And in fact, polyomaviruses were found to cause tumors in 1953 by Ludwig Gross. I love this. Look at this guy, all these mice here, old wooden cages. Wow, so cool. All right, so a murine polyomavirus. It caused rare tumors under certain conditions. The natural host is the mouse. And these viruses are ubiquitous in mice. If you trap wild mice, you'll find lots of polyomaviruses, but they don't cause cancer in mice. They cause cancer in the wrong host. They make many tumors of many tissues. That's what polyoma means, many tumors in hamsters, rats, and rabbits, but not in mice. So that's a very important point, the wrong host. Then we have SV40, another polyomavirus. Bernice Eddy and Maurice Hilleman discovered SV40 as a contaminant of the early poliovirus vaccine. SV40 stands for simian virus 40. We use monkey kidney cells to grow poliovirus vaccines. And in the early days, we didn't realize that you had to check for other viruses, and some of the early lots had this contaminant SV40, but this causes rare tumors in newborn hamsters. They showed this in 1962. And in fact, several million Americans were infected with this virus, along with poliovirus. If you look into the literature, you'll see claims that this causes tumors, but I don't think it does. I don't think the data are correct. I don't think it does anything in people. Natural host of this virus is the monkey. That's why it's called simian virus 40, but it doesn't cause any tumors in monkeys. And it doesn't transform monkey cells in culture. But it will transform hamsters and cause tumors in hamsters. So again, that's a theme here that we're going to consider, the wrong host. So this is just a summary of all this. So we have species, and then we have the virus, SV40 or mouse polyomavirus. So if you take a monkey and you infect it with SV40, the monkey is both susceptible and permissive for the virus. So the virus replicates, no tumors. The virus is killing the cells. If you infect monkeys with mouse polyomavirus, it's completely non-permissive. The virus doesn't replicate at all, so nothing happens. You don't get any tumors. If you take a mouse, you infect it with SV40, the cells of the mouse are non-permissive, no tumors, no replication. Mouse polyomavirus in the mouse is permissive. It destroys cells, so no tumors are formed. But the animals where you get tumors, the hamster and the rat. In the hamster, SV40 is semi-permissive, and mouse polyoma is semi-permissive. Same thing in the rat, semi-permissive, semi-permissive, and that's the key. I'm going to explain that in a few slides. If you're non-permissive, no tumors because the virus doesn't even have a chance to replicate. If you're permissive, the virus kills the cells, but semi-permissive is the key. And what that is exactly, we'll explain. The transformation of cultured cells by polyomaviruses is rare. You get about 1 transformed cells per 100,000. You take a monolayer of cells and you infect all of them with polyomavirus. Only 1 in 100,000 are transformed and give these foci of transformed cells. So I want to answer for you in the next few slides, why is it rare and how does this relate to rare tumor formation, say, in animals? Finally, another family of viruses that transform cells, adenoviruses, which we have talked about a bit in this course so far. There are many human serotypes. None of these cause cancer in humans. No human adenovirus causes cancer in humans. However, some will cause tumor in hamsters. 12 to 18 are causing tumors, and 7 to 11 are poorly tumorigenic, but they will cause tumors in hamsters. And like polyoma and papilloma transformation in tumors, they're very rare events, and they're in the wrong host. Humans don't get tumors after adenovirus infection. And the key that links all of these DNA viruses together are T antigens. And T antigen we looked at way in the beginning for SV40, and T stands for tumor antigen because it was a protein found in all the tumors. And for all these viruses, SV40, polyoma, papilloma, adeno, they could always find a protein in tumors. They called them T, and they're all different. Now here's a map of the SV40 large T, and so SV40 actually has 2 that are both present in tumors. Polyomas have a large, middle, and small T. Papillomas have a T encoded by the E6 and E7 genes of the virus, and adenoviruses, the T antigens are the E1A and E1B proteins. So what you need to know right now is that all these proteins are found in tumors and transformed cells, and for each virus, the protein is a different protein. You'll see how they do this in a moment. Now remember, for SV40, you know that T antigen is an essential gene. T antigen of SV40 is required for replication. It activates transcription. It's needed for viral DNA synthesis. The large T binds to the origin and recruits the DNA synthesis machinery. And the key here, these are the only viral genes always retained in tumor cells or transformed cells. So if you take any of these viruses and you infect cells in culture, you get transformed cells, and put those in an animal and get a tumor. The only viral proteins that are in all of them are the T antigens. And in fact, if you take the gene for SV40 large T on a plasmid and put it into any primary cell, you can transform it. That's how you can make cell lines in the lab. If you wanted to make a cell line from you, if you want it to be immortal, you take a few cells from your cheek with a Q-tip, put them in culture, let them grow a bit, put a plasmid encoding T antigen, they'll grow forever. Can be immortal. Finally, to link all of this together, I have to tell you 3 discoveries which were made independently. They seem unconnected initially, but they were the link between these viruses, these DNA viruses, transformation, and the cell cycle. First, people studying SV40T found it bound a cell protein called, well, we now know it's P53, but it was a 53,000 Dalton cell protein. It just bound it. 2, transcription of a set of adenovirus early genes, the E2 gene cluster, required a cell protein called initially E2 factor. Now we know it's a family of transcription proteins called the E2F family. So apparently the adenovirus needs this, right, for the transcription of early genes. And finally, E2F, this E2F transcription factor was found to bind a cellular protein called retinoblastoma protein, or RB. And P53, RB, and E2F were all connected and found to be critical in the control of the cell cycle. So now we have to talk a little bit about the cell cycle to set this up for you. As you know, every cell divides around 24 hours. Most cells divide around 24 hours. And there is a distinct phase of mitosis, or M, where the cells actually split apart. But after the cells have divided, there's a period of cell growth called G1. There's a period of DNA replication called S. And then just before cell division, a G2 period. So just before the mitosis period, cells can be resting. And when they're stimulated to divide, the cells then go from mitosis into the G1 phase, and then they replicate their DNA. So this is a tightly controlled process because, as we said at the top, you don't want cells to divide always because, first of all, you don't need it, and that would accumulate mutations and the cells would become transformed. And so this is the control that we have learned from studying transforming retroviruses, and that is whether a cell proceeds through mitosis is determined by nutrient concentration and growth factors. There are receptors on the cell surface for growth factors. If there's enough in the medium, they will bind those receptors and transduce a signal to push the cell through the cell cycle. Another way would be to look at this is the outside world rich enough to replicate the cell? And these growth factors, the receptors, the whole signaling pathway that turns on cell division, were discovered as oncogenes carried by transforming retroviruses. That's one of the amazing outputs of this story. This whole growth control pathway was discovered by studying these transforming retroviruses. Now, there's still another checkpoint on this whole cell cycle control. Let's go back to the beginning. We have mitogenic signals like growth factors binding receptors. In order for the cell to proceed through mitosis and G1, RB has to be dephosphorylated. Now, RB is a protein encoded by a gene, stands for retinoblastoma protein, that was originally discovered in kids who have retinal tumors. And these are young kids because the cells are gone by age 5. So the tumors were detected in young kids, and they were found to have missing both copies of this gene, which they called RB, because it was associated with retinoblastoma. It is a recessive oncogene. You take away both copies, and now you have tumor transformation and tumor formation. And RB regulates these two checkpoints. At the onset, if you have mitogenic signals in the form of growth factors delivered to the cell, RB will become dephosphorylated, and that lets the cell proceed. And as you know, the level of cyclins in the cell rise in different parts of the cycle, G1, S, and G2, and they eventually lead to the distinct phases and eventually mitosis. If the conditions are not right for the cell to proceed through the cycle, RB will become phosphorylated, and that will stop the cycle right here at this restriction point. And if you never go through S, you will never have cell division. So that red line, that restriction point, kicks in when conditions are not right. We'll see in a bit what those conditions would be. But again, this is a protein discovered in retinal tumors because the kids lack the gene for both copies of the gene. They don't have RB, so the cells just kept dividing. There was never a restriction point in those cells, and they became tumors eventually. So here's how this works at a molecular level. On the left is our mitogenic growth control pathway. We have a growth factor receptor. So the ligand is a growth factor, which when it binds to a growth factor receptor, will start a signaling pathway that leads to mitosis eventually in the nucleus down here. All of these components, the ligands, the receptors, the signaling proteins. They're all genes identified in transducing retroviruses. And what happens here is when the growth factor binds signaling, activates a variety of kinases, cyclin levels go up, RB becomes phosphorylated. That leads to mitosis. Now, you should remember why this causes the cells to divide, but I'll remind you it's on the right here. So normally in cells, RB is bound to E2F. They're bound to promoters. They recruit histone deacetylases and keep the promoters off. So these cells cannot divide because these promoters control the production of cyclins and other proteins needed for DNA synthesis and ultimately mitosis. When RB is phosphorylated, it pops off of E2F, and that activates these promoters. You can have cyclins made. You can now go into DNA synthesis. So it's essential for RB to be phosphorylated to go from G1 to S. Remember that DNA viruses need cells to be in S phase so they can replicate. And we talked about this with T antigen. We said T antigens kick quiescent cells into S phase. So, if the conditions are right, the cells will go through mitosis and proceed into G1. But if conditions are not right, RB will be dephosphorylated. You will not get progression through the restriction point. Remember, we have to have RB phosphorylated to pop it off E2F. And T antigen, at least of SV40, is what I showed you earlier in this course. They can kick quiescent cells into S phase, and we talked about this earlier. So remember, phosphorylation of RB can knock it off of E2F, and then the promoters become active and make the proteins you need to get DNA synthesis and mitosis. T antigen, large T, E1A of adenovirus, and E7 of papillomaviruses. They're shown in purple here. They will also bind RB and pop it off of E2F, thus activating the mitogenic pathway. Now remember, adeno also requires E2F for transcription of its early genes. That was the link between all of this, or one of the links between all of this. So a cell can regulate whether it goes into cell division or not by phosphorylation state of RB. And viruses can bypass all of that by simply binding RB with their T proteins, the only protein that's present in all tumors. So when viral T antigens bind RB, E2F are released and initiate S phase transcription. However, there's one more control over the entry into S, that DNA synthesis phase. So let's say that the T antigens have bound RB and now the cell is proceeding into DNA synthesis. There's one more checkpoint that cells have there, and that is P53. That protein originally identified because it bound T antigen of SV40. When a cell begins to do unscheduled DNA synthesis, that is viral DNA synthesis, that's not scheduled by the cell. P53 can detect that. When there are double-stranded DNA breaks, which happens in virus and replication, SV40 has to break its DNA to untwist it. That's detected by cell proteins, and that activates P53. P53 then binds to a promoter, a variety of promoters of what are called P53 responsive genes that lead to cell cycle arrest and cell death. So the cell doesn't want unscheduled DNA synthesis, doesn't want damaged DNA. Don't duplicate damaged DNA. And P53 controls that. So if a virus is going to kick a cell into mitosis, it has to counter P53. And here's how they do it. These T antigens all counter P53. In addition to binding RB to get past that checkpoint, they have to counter P53. So here's P53 here. The E6 protein of human papillomaviruses, they bind P53 and cause it to be ubiquitinated, which causes it to be sent to the proteasome for degradation. Large T of SV40 and the E1B protein of adenoviruses, they both bind to P53 and sequester it so that it cannot activate the genes that lead to apoptosis. So viruses counter P53 in addition to popping RB off of E2F. Otherwise, the cell would never divide. All right, our last question. T antigens are A, encoded by viral genes that are essential for replication, B, present in tumors and transformed cells, C, encoded by viral genes that have been incorporated into the cell genome. D, antagonists of cell cycle checkpoint proteins, all of the above. All right, how do we do here? All of the above is right. T antigens are encoded by viral genes. They're essential for replication. They're present in tumors and transformed cells, encoded by viral genes that have been incorporated in the cell genome, and they're antagonists of cell cycle checkpoint proteins. Two more mysteries. Why are all viral genes except T antigens deleted or turned off in these transformed cells? And why is transformation so rare? So we're going to answer that. It's rare because of two low probability events that have to occur. First of all, the late genes cannot be expressed because they're lethal. And so when a virus infects a cell, in some cases there's a rare spontaneous deletion of the late genes, and that will prevent the cells from being killed. So the late genes typically encode capsid proteins, and you accumulate virus particles that's going to kill the cell. So you can have rare spontaneous deletion of late genes. So it's pretty rare. It's a low probability event. And then you have to infect semi-permissive cells. And the definition of semi-permissive, which I promised to give you a long time ago, means late expression. Late viral gene expression is blocked. That's what semi-permissive means. So if you infect a semi-permissive cell, you never get to late, so the virus doesn't kill cells, better chance of causing transformation. However, you need one more thing. You need T antigen on all the time. It has to go to every cell as it divides. That's how a transformed cell propagates and becomes more transformed cells. So the DNA encoding T antigen has to be integrated into host DNA. So you have to suppress late genes and you have to integrate the DNA. Those are both very low probability events. That's why a transformed focus is very rare when you infect cells with these viruses. And using the wrong host makes the infection semi-permissive. That's why they transform just in the wrong host. In the normal host of SV40, polyoma, for example, adenovirus, they just are lytic replication. They kill the cell. There's no transformation potential. So by understanding all this, we realize that transformation and eventually tumor formation are abnormal processes for these DNA tumor viruses. They're abnormal epigenetic processes. They're outside of the genome. They're something that's happening in the cell. They're not required for normal viral reproduction or transmission, except in the case, of course, of walleye, but that's not a DNA tumor virus. And they were discovered by infecting the wrong hosts. So people just took these viruses and they infected different animals in the lab and they saw tumors. And if they weren't curious in that way, they never would have found this. Given all this, we now know that even in the natural host, sometimes these rare events may occur and cause tumors, and human papillomavirus is an example, and I'll come back to that just at the end and explain that. So transformation is an epiphenomena of a unique reproductive cycle. An epiphenomena is a secondary effect that arises from, but does not causally influence a process. So it arises from the way these viruses replicate, but does not influence that replication. For the case of DNA tumor viruses, they have to kick the cells into mitosis in order for their genomes to be replicated. And so T antigens turn on the cell cycle by inactivating RB, but that's not enough. They also have to block P53. So these T antigens would normally just be doing that to allow lytic virus replication. But if you have two rare probability events, the wrong host and integration of the T antigens, then the cell gets transformed. And so if you put these T antigen genes into cells, the cells becomes transformed. And if they replicate enough, they will become cancer cells. So if you make a primary line and then put a T antigen in it, and then immediately put it into a nude mouse, you may not get a tumor. But if you let it divide for a couple of months and sustain mutations, then you put it in a mouse, you've got a tumor. Okay, now we can explain HeLa cells and Henrietta Lacks' cervical cancer. Remember, HeLa cells were derived from Henrietta Lacks in the early 50s. She had a cervical tumor. Her doctor took out a piece of it, chopped it up, put it in culture, and the cells lived forever. The first immortal human cell line. Back then, they had no idea why it was immortal. But now we know, because not too long ago, the genome sequence of HeLa cells was done. And you can see integration of human papillomavirus type 18 in one of the Lacks chromosomes, number 18. The only intact genes. This, by the way, HPV-18 is one of the high-risk papillomaviruses. There are many papillomaviruses that cause warts of all kinds, but a few of them are high risk for causing cancers, and this is one of them. So she acquired it, probably sexually at some point in her life. And something rare happened in her case. The DNA integrated, and only E6 and E7 genes are left. There are pieces of some other viral genes you can see in the HeLa genome, but these are intact. E6 is the protein that binds RB, and E7 degrades P53. And that's why she developed a tumor, because the DNA aberrantly integrated. This integration is quite rare in infected individuals. So everyone who's infected with a papillomavirus is not going to have DNA integration, but occasionally it happens. If, in fact, you only have E6 and E7 genes present and the rest of the genome is not there, so it doesn't kill the cell, then the cell would become transformed. And after enough divisions, the transformed cells will sustain the 12 or so mutations you need to become a cancer, and then a tumor develops. And that's why Henrietta Lacks died, because of that tumor. And that's why HeLa cells are still to this day growing in laboratories. Here's a summary of everything today. We know that transformation by these viruses, both RNA and DNA tumor viruses, these are epiphenomena of unique reproductive cycles. In the case of RNA tumor viruses, the viral DNA has to integrate. And when that goes in the wrong place, it can pick up an oncogene or activate it. And in the case of DNA tumor viruses, they have to turn on the cell cycle in order to replicate their DNA. And sometimes, in the absence of a lytic infection, that can transform cells. So they are accidents, essentially. And now we understand this entire control of cell growth. We know what turns on mitosis, the go signal at the top, by studying the transforming retroviruses that have the dominant oncogenes that encode all the machinery to get the mitosis started. And we also know all about checkpoints within G1 and then within S. Those are the tumor suppressor genes, the recessive oncogenes, which we see by studying DNA tumor viruses. And this all started with Rouse in the early 1900s. So his discovery of an RNA tumor virus, a retrovirus that caused tumors in chickens, then spurred other people to looking for DNA viruses that cause tumors as well. And so the really remarkable story here is, of course, all of this started with a chicken and a tumor. So next time we're going to do something very different. We're going to take 2 lectures and talk about how to prevent and resolve virus infections. So we'll start next time with vaccines, how to make them and how they work.